So good morning, everyone. Great to see so many faces in the morning after the many social events that were going last night. So today, as you've noticed from the title, we'll be having a couple of talks, and we are happy that we have 3 of our presenters here in person. Dani Behjat, she has apologized, she couldn't attend, so we will just play her video. But even on her talk, if you had questions, please post your questions on the app, and then I will reach out to her, and responses will be put up later in the app for those who are interested. And even in the app, there is two little small polls that were created. You can even log into there, and if you're interested, to just respond to those. And please do, if you have any questions during the talk, just put it up in the app so people can vote it, and then we have a nice discussion. Okay, so with this, I would like to introduce Katharina, who's a postdoc at the Berlin Institute of Health at Charité, and she will be showing us how GSP principles can be used on functional connectomes. So basically, purely from a functional perspective, but how these can be used to understand some organizational principles from the human brain on fMRI data. So, please, Katharina.
Thank you so much. And thank you to the organizers for giving me the opportunity to speak here in this, frankly, shockingly big room. Thank you for coming here this early in the morning. So I'm going to do my best to give you a brief teaser of what you can do with so-called functional harmonics and what they have to do with cortical organization. So to start, I just want to say that all of this wouldn't have happened without Selen Atasoy, who was a postdoc in the same group where I did my PhD, and she first asked me if I wanted to work on this topic, and this is where this paper, the figure of which you can see on the right-hand side, came out of. So very brief. I don't have time to go into details of the mathematical stuff about functional harmonics, but just imagine we're working with a brain graph where each node is a brain region and the connections are, in this case, functional connectivity, so correlations. And this is a really strange domain on which we want to analyze our data. But in the end, it's just a domain, just like when we analyze the usual 1D continuous signals. In that case, the domain is a circle. And we are all familiar with Fourier transform, where we use sine and cosine waves to decompose our signal into these simple components. And suffice it to say at this point that functional harmonics are nothing else but the corresponding thing to sine and cosine waves on the domain of the circle, but applied to the graph. And I'm going to show you what that means in practice. So apart from this very hand-wavy principle, the way that you compute functional harmonics is actually very easy, very simple. I can only recommend, if you're interested in this topic, to just try it out. In this case, in this paper, we started with HCP data. I know we've heard a lot about how we're biased towards this data set, but well, we used it too, because it's very nice data, very high quality, and it's 812 subjects in the case that we used. And you can actually download the dense functional connectivity matrix directly from their server. So dense means that we use the entire surface mesh with 60,000 vertices, so no parcellation in this case. And what we did then was we just simplified this a lot by looking for the 300 nearest neighbors, and we created a sparse binary functional connectivity from this. And from that, it's very easy to compute the graph Laplacian, which is this fancy sounding thing with the fancy squiggly G. So it's the graph Laplacian. And you do this, you compute this very easily by just subtracting this sparse binary FC, in this case the A, from this matrix D, which is nothing else but a diagonal matrix, which is 0 everywhere except in the diagonal where it has the number of nearest neighbors, so around 300 in our case. And then you apply an eigendecomposition, which is, again, is an algorithm that is very easy to apply. I mean, in the case of a 60,000 by 60,000 matrix, it may take you some time. You can't maybe compute all the eigenvectors, but we computed the first 100 and looked at that. And then these eigenvectors are the functional harmonics. So they are, in a way, a low-dimensional embedding of the functional connectivity matrix. And because it's an eigendecomposition, it follows that because we have n vertices, so in this case about 60,000, we also have about 60,000 eigenvectors in theory. So there exist about 60,000 harmonic modes. Here I'm just showing you the 11 first, so a tiny fraction of those. We analyzed those in the paper because we found that they were the most relevant for task activity patterns. And you can see these beautiful patterns that range from very coarse patterns that kind of show very big parts of the cortex in the same network, let's say, to more finely grained patterns when you go to higher order eigenvectors. Okay, how to interpret this, what to do with these nice looking but useful question mark patterns. So one thing to understand here, I already mentioned, these are low dimensional embeddings of the FC, and each of the functional harmonics can be interpreted as a separate embedding. Of course, if you want to understand cortical organization, you kind of have to see them together, the space that they span, but you can look at them separately. That's what we did. And they are smooth patterns on the cortical surface. So we just projected the eigenvectors back to the cortical surface, and you see these smooth patterns with these varying colors that, of course, correspond to the continuous values in the eigenvectors. And the way to interpret this, to read this, is that the same color signifies a similar FC pattern. And if you make a little jump, you can interpret these as networks, of course, because the same color marks vertices that have similar functional connectivity patterns to the rest of the cortex. And here, I'm just showing the first two, and many of you will be familiar with this picture. I'm just showing the one from this wonderful paper from 2016 from Margulis and others. If you look a bit more closely, you will see that we reproduced this perfectly, so it's exactly the same. And the way to interpret these two gradients or these two functional harmonics is that they are large-scale gradients on the cortical surface that define this kind of triad between visual network, sensory motor and auditory networks, that's in green in the picture on the bottom left, and then higher order regions, or as Margulis et al. Showed, this is mostly DMN regions. And the point here is that this is not like three separate clusters where we're pretending that the brain is subdivided into disjunct networks, but you can see the spread of the points of the vertices between the three points of the triangle, and how this maps out a gradient and a smooth transition between these networks, between these functions. Okay, and now something very important about these eigenvectors. So as any eigenvectors, they are ordered by eigenvalue. And in this case, the interpretation of the eigenvalue is not, as you might be thinking if you're now thinking about PCA, is not a direction of highest variance or something like this, but it's actually smoothness on the surface, or in other words, it's increasing frequency on the graph. So this is why I mentioned earlier that you can see bigger networks in the top rows, so in the first few eigenvectors, and then as you go further down, you see smaller patterns. So this is like the increasing frequency of sine and cosine waves that I mentioned earlier for the circle. It's exactly the same. Of course, it's not as easy to spot because the graph that we're talking about is much more irregular. Yeah, there's a clear definition of graph frequency in this case. So you can see finer subdivisions of the same network. And I'll show you what this looks like in one of my favorite examples. I apologize to everyone who has seen this time and time again. So if you start with the second eigenvector in the top row, you can see, I don't know how well it's visible, but like, oh, is this a pointer? Nope. Okay. So you can see this big somatosensory somatomotor network in blue or cyan. And then if you go to a slightly higher order one in the seventh eigenvector, you can see the subdivision of this same network into somatotopic regions. And here you see these glowing yellow regions, and those correspond to the left and right hand. So it makes sense that on some level you have common connectivity of the entire huge network, but at the same time, of course, the different parts of your body can do different things and feel different things. So it also makes sense to have a subdivision here. And the 11th eigenvector was actually the first one that we observed that was really asymmetrical. So in this case, you can see this opposition of the left and the right hand, which again makes a lot of sense. Your hands are similar in a way, but they can also do things separately from each other, and they have different functions. So this is how you can use functional harmonics to study integration and segregation, let's say. Okay, and another feature that I find really nice about using harmonic modes or functional harmonics is that you can understand how one region contributes to multiple networks and how it can have different functions depending on the context. Of course, it won't tell you what context that is, but you know, baby steps. So if you just use seed correlation, for example, so here we have area MT, also known as V5. It's known to be involved in motion perception. So that's where this arrow points to in the top row. And if we look at three different functional harmonics, so again, three different eigenvectors with different graph frequency, you can see that this region is grouped, so similarly colored with different groups of other regions. And you can, I mean, in this case, we could identify quite clearly the kinds of networks that we had. So this region has something to do with audiovisual integration. If it communicates, let's say, with auditory network, it's also involved in the sensory motor pathway, which we know from other papers. And it's also involved in the ventral stream, which you can see here, especially from the contribution of the fusiform phase complex. Okay, and now I'm going to come to my last point, where I want to say a few words about the more mathematical part of these functional harmonics. So as I said, they are eigenvectors, so you can use them as basis functions. And to understand what I mean by that, I just want to emphasize something that should be obvious, but sometimes is hard to keep in mind. So the graph itself from which we extracted the functional harmonics doesn't change. That's our domain. But then on this graph, we can have a time-varying signal. We can have different activity patterns, right? So there's a separation between the graph or domain and the signal, in this case, the graph signal. So what you can do with functional harmonics as basis functions is that you can decompose your graph signal, in other words, your brain activity pattern, into basis functions, exactly like you would do with a one-dimensional signal, like a time series or whatever kind of physical wave pattern. You can decompose it into sine and cosine waves. In this case, you use the functional harmonics as basis functions. So the Fourier transform becomes the graph Fourier transform, and it's exactly the same thing. And you can do all the things with it that you can do with the Fourier transform. So you can filter, for example. But you can also characterize activity patterns on the cortex by their harmonic modes spectrum, just like you would characterize a signal in time by its spectrum of oscillatory frequencies. And that's exactly what we did. We took 47 task maps from these 7 well-known task categories from the HCP. And in the next slide, I'm just going to show you 3 examples, and you will see how different they are. So on the left side, there will always be the original activity pattern. On the right side, you will see what happens if you add more and more functional harmonics to reconstruct this pattern. And so the bottom row is basically the best. So if you zoom in a bit with your attention, you will see the bottom row on the right side looks a lot like the activity pattern that you see on the left side. And then the fancy way of showing the spectrum in this, I forgot the name now, in this round spectrum in the middle just shows the activity or like the contribution of the first 11 functional harmonics that I showed you before. So here, it's a very busy slide, but I just want you to appreciate the difference between the spectra in the middle. So these are different tasks from different task categories. So you can see that the contributions of the functional harmonics are very different in the memory task versus the two motor tasks, and that the hand movement task is very different from the tongue movement task. So you can reconstruct all these patterns, and so you have now reduced your 60,000 vertex pattern to 11 functional harmonics. So that's a one-dimensional thing, right? Okay. So I'm almost at the end. So as a summary, functional harmonics are eigenfunctions of the resting state functional connectivity brane graph, but you can compute harmonic modes from other graphs, of course. They embed the FC into a row of one-dimensional representations over a range of spatial frequencies. And they reflect several organizational features of the FC, of which I have shown you hierarchical networks, global connectivity gradients, and integration and segregation. And activity patterns can be understood as a superposition of these functional harmonics. Okay, open questions that you can also use as inspiration to ask me questions or ask questions later. Is there lots of methodological questions that we still have to answer? We're trying to figure out how to use these harmonics to explore differences in network organization between individuals, groups, or different brain states. Yeah, because they look nice, but what can we do with them? And what I'm personally very interested in is whether there is a connection between the functional harmonic spatial patterns and oscillation frequencies, like we would have with standing waves. Yeah. And with that, I just want to thank once again all the co-authors on this paper, and thank you for listening.
So thank you very much, Katharina. So we have a couple of questions. So the first one is, I think you touched upon it a little bit, but the question is, in what sense do functional harmonics differ from so-called gradients, functional gradients?
They are the same thing. It's just a difference in interpretation or in approach, let's say. So the people that I work with, we mostly think about it in terms of graph signal processing, dimensionality reduction, and there's other people that do amazing work that think about it more in terms of organizational gradients and connect it to, for example, microstructural gradients and things like that. So. But in principle, it's the same thing.
Okay, and next question is, how much of the variance in the data is explained by the first 100 functional harmonics that you mentioned?
I don't have the numbers in my head, but let's say it differs depending on the pattern. So, for example, for the motor tasks, we have functional harmonics that represent those very nicely. So even the first functional harmonic will already capture a lot of the variability. And for other patterns, for other things that are more cognitive, let's say, you need more functional harmonics to really capture the activity pattern. And I mean, also keep in mind that these are group averages. So, yeah.
So that links me to the next question. So how much do group-wise functional harmonics differ from subject-specific functional harmonics? So basically, is there some fingerprinting power in the functional harmonics?
So we're only beginning to explore this, or I am at least, and I can say that the first few functional harmonics are quite stable, also if you look at individuals. But already, if you look at, like, I would say around the 6th functional harmonic, you already see big differences between the group and the individuals. And yeah, I'm not sure yet if that really reflects a difference in people's brains, or if that is a problem with our methodology, and I think it's probably the latter.
Great. So thank you very much, Katharina. There is a couple more questions. Maybe you can check out the questions later on, but we have to move to the next speaker. So our next speaker is Giulia Preti, which together we are organizing this session. Giulia will be presenting her work on, again, using graph signal processing, but she will be giving us a different perspective. So basically, in the work that Katharina presented, functional harmonics are purely derived from functional data, but Giulia will show us how you can derive harmonics or these bases from structural data and then map functional data onto that basis and make interpretations. And Giulia is a researcher at CIBM in Switzerland and also a lecturer at the University of Geneva. So please go ahead.
Thank you for the introduction, and thanks all of you for being here at our early morning symposium. So congratulations on having made it at 8 a.m. So I'm very pleased to be here today to talk about my work on graph signal processing for exploring the relationship between structure and function in the brain. So there are a lot of resemblance with what Katharina already greatly explained, but in this case we are looking at structure and function together. So you will see this is the main difference. So the work that I will present, the results are mainly contained in this paper that we published recently about indeed the fingerprinting potential. So I mean we will talk about that, and also task decoding potential of these measures. But I will be referring a lot to the first original paper where we introduced these metrics to quantify structure and function coupling in the brain. So these are all the details, since the talk is quite short, for you, in case you're interested. So to give you a little context, as I said, we want here to combine the information about brain function and structure. So the data that we're using comes from MRI, and you already know for sure. So the functional information that we're referring to is the fMRI functional activations, and the structural information comes instead from diffusion MRI that allows you for a virtual reconstruction of the white matter paths underneath. So combining these two is non-trivial because they come from different acquisitions, and it was shown already in the past that there is not an easy one-to-one relationship. And so graph signal processing in this context helps you to integrate the two, and it's a new emerging framework that was only recently applied to the brain, and it's very useful for that, as you will see. But how does it work? So we need a graph, yes, and for us, contrarily to the work of Katharina, the graph is a structural graph. So it's structural connectivity, where nodes represent region in the brain of a certain parcellation, and edges quantify the connectivity, the structural connectivity between different regions. And then on top of this graph, we define a signal, which is called a graph signal, because it has the same dimensionality of the nodes of the structural graph and is really defined on top of these nodes. And the signal for us is the functional activity, so the bold fMRI activity at each region at one time point. So it's very important here to remark that normally we are used to think of fMRI signals like time courses. Here instead we are working in space, in the space, nodal space of the graph. So basically a signal for us is a snapshot of the brain in terms of function. And then for the nature of fMRI, you all know it varies in time and we acquire multiple time points. So we will have multiple signals. So we'll apply the method at the different time points then. So the question that we can address with this method is, how is the function using the underlying structure? How do we do it? So first, we decompose the structural connectome analogously at the work of the first presentation that you saw. We used Laplacian eigenvectors, but in this case we obtain structural harmonics, so patterns that sum up the structural connectivity. Each of them is associated to a level of spatial frequency which corresponds to lambda, so to the eigenvalues. As Katarina already showed, so this eigendecomposition yields eigenvectors that maximize smoothness on the graph. So we have increasing value of spatial frequency that you can see here as an example. So let's see how structural harmonics look like, because they're very different from functional one as they reflect structural connectivity. So the first one is spatial frequency 0, okay? Eigenvalue 0 corresponds to constant pattern, no variation across the brain. So we have our constant pattern throughout the brain. It's a bit the DC of your decomposition if you think about it in signal processing terms. And low frequency eigenvectors, so the ones at the left hand of the spectrum, they're very smooth on the graph. So they are characterized by a very low value of spatial frequency. You have very smooth variation, and meaning that here, what you see with the same color really corresponds to nodes that are very connected in the structural connectivity. And in fact, you can see that we can capture the main axis of the structural connectome, so the difference between left and right hemisphere, for example, or anterior to posterior gradient, or a mix of them. But a feature that really all these patterns, low frequency patterns, have in common is being global patterns and being very smooth on the graph. So they are not that distant from the structural connectivity. In other words, that will come after, they are coupled to that. If we move instead to the right-hand side of the spectrum, so we look at high spatial frequency structural harmonics, these are patterns that instead have a very high embedded distance with the graph. So they are not as smooth as the first ones on the graph. And you will see here a lot of focalized patterns and a lot of red-blue switch. So indeed, these patterns are more decoupled from the graph that we are considering. So understanding this difference in the decomposition is very important. So here, if you want, we are not only interested, it's not a dimensionality reduction, so we are not only interested in the first eigenvectors, but we are interested in the whole spectrum, because our interest is indeed characterizing different structural harmonics based on if they are coupled or decoupled to the structural connectome. And it's very important to understand this difference, because now then what we do is, as a second step, projecting functional activity at a certain time point on top of these structural harmonics. So in other words, we explain each pattern of functional activity as a linear combination of structural patterns. By estimating the coefficients of the linear combination, this has had coefficients that you see here, we explore what is the contribution of each structural pattern at each time point in the functional activity pattern. So this is an equivalent of the Fourier transform in conventional signal processing terms, because as you all know, in the Fourier transform, you you take a signal from the time domain to the temporal frequency domain. Here, instead, we go from the space of the graph, so from the nodal domain to the graph spectral domain. So we decompose the graph in its spatial frequency in terms of structural component in this case. Then what we want to do is, in the spectral domain of the graph, filter these signals. So we want to decompose our functional activity signal S1 here into two portions, one that only includes the low frequencies and one that only includes the high frequencies. So we basically apply simply ideal high-pass and low-pass filters with a certain cutoff. And what we get is when we reconstruct back the signal, it's two portions of the signal that, if summed up, they give again the functional pattern. One that we define as coupled, because it only includes structural harmonics that were very smooth on the structural connectome, and the other that is indeed more decoupled. So this is a real example. So you see that an fMRI frame of a certain subject is subdivided into these two portions, that the coupled portion captures more the portion of functional signal that is directly explained by structural connectivity underneath, while the decoupled portion captures the patterns that ride on top of it. So whatever is left, basically, in the signal. So we do that for each time point. We repeat that for time point 1, 2, 3, and so on. And what we obtain is two versions of the time courses now, okay? One that is coupled to the structural connectome and one that is decoupled. And then we can ask, are there regions in the brain that along the acquisitions are most often coupled or decoupled to the structure? So is there a spatial pattern of structure function coupling? And so to answer this question, we computed the L2 norm across time of these two time courses, coupled and decoupled, to obtain two brain patterns, one that quantifies coupling and one that quantifies decoupling with the structure. And then the ratio between these two measures, so the ratio between these two patterns, yields what we call structural decoupling index, which is a quantification of the structure-function relationship at the nodal level. We can also measure structure function coupling at the edge level, a bit like what we do with functional connectomes, connectomes in general, because we also wanted to compare it then afterwards in terms of fingerprinting and task decoding with functional connectivity. So what we do here, okay, exactly as you compute functional connectomes for functional time courses normal, we computed Pearson correlations, pairwise Pearson correlations, but starting from the coupled and decoupled time courses to obtain these two versions of functional connectivity, one that is coupled and one that is decoupled. So these are the measures that we'll be using in the following. So to recap, you have now the full picture of the method. So first step, you decompose a structural connectome into structural harmonics. Then you project your functional activity at a certain time point into this space spanned by the structural harmonics. You filter them into a coupled and a decoupled portion, and then you quantify structure function coupling at the node level and at the edge level with these metrics that we introduced. But now, how does that look like? So for healthy participants of the Human Connectome Project and for resting state, on average, this is the pattern of structural decoupling index that we found. So first, we can notice that it's not constant across the brain, right? And it was very interesting to see that, indeed, it spans a very meaningful gradient, going from regions that are more sensory, more primary sensory regions that for us are coupled to the structure, the ones you see here in blue, to regions that are more decoupled that happen to be the one responsible for higher level cognition. So multisensory integration, decision making, memory, and so on. So this gradient has indeed a behavioral relevance that we confirmed also with the neurosynth topic term analysis, where indeed the regions that are more coupled, the ones that you see here in blue, corresponded more to lower level sensory functions like motor, eye movements, and so on, while the regions that are more decoupled corresponds to more complex tasks, let's say, and higher cognitive level tasks. And these results replicated the very famous ones on functional gradients introduced by Margulies in 2016. So we really wanted to perform the same analysis here to see if there was a correspondence. And what we are adding is, okay, these gradients, you can find them also when you're looking at how structure and function relate, basically. And then in terms of interpretation, we found strong congruence. So these gradients are present in many other studies based on different things that you can look in the brain. For example, one that I find very interesting is a congruence with a genetic study that found very similar networks, the one that you see here in blue and red, but in terms of which proteins are used for neural transmission. And the coupled network, so what we found out to be a coupled network, seems to use more genes that favor a faster response, so these primary sensory networks, so they have to act fast. And what we are saying is, yes, so the functional signal in that case follows directly the structure underneath. While for the red network here, you have genes that favor a slower, more sustained activity, and we are saying these are the decoupled areas. So you probably don't have such a temporal constraint if you are doing more complex tasks, thinking about your past, and function here can also afford basically following indirect path of structural connectivity. But okay, all this was related to a group level organization on healthy subjects and during resting state. So do these measures of structure function coupling also characterize different subjects, or do they characterize tasks? What happens when we are doing a task in the scanner? So in order to answer these questions, basically we wanted to see if there was a potential in these measures for fingerprinting and also for task decoding. And in order to answer these questions, we analyzed another set of HCP subjects with the 7 additional tasks that are available apart from resting states. And then we tried to classify subjects and tasks. So here are the results. So accuracies for classification, we have adopted a support vector machine classification with different cross-validation settings with respect to if it was for fingerprinting or task decoding. Accuracies are very high. What we can notice is that for task decoding, at the nodal level, so we tested these 5 measures, so both at the edgewise and node-wise structure function coupling measures, but also we wanted to compare it with functional activity, basically, functional connectivity. So we see that at the nodal level, the SDI outperforms the functional connectivity node strength for task decoding accuracy, and accuracies are very high instead for all the edgewise measures for task. But also what is interesting to notice is that for subject fingerprinting, accuracies are very high, but the highest one, perfect accuracy of 1, is reached with DFC, so with the decoupled version of functional connectivity. And this is very interesting for us because, we don't really know what is this signal, right? What portion of the signal goes in this portion, the decoupled one. And so it's interesting to see that it's indeed the portion of the signal that is more unique to different subjects. And this was confirmed also by this other more difficult cross-validation setting when we are trying to predict one task using only one other task, and then applying them for all pairwise combinations. And even here, we see that functional connectivity works very well. So we already know about the fingerprinting potential of functional activity in connectivity in literature. But we wanted to see if it works also for a structure function coupling. And what we found here is that it doesn't work well for coupled portion of functional connectivity. So it's more, when you decompose the signal, it's more in the decoupled portion that you find this information that is relevant and useful to identify uniquely subjects. And the relevance, the importance of this information was also confirmed by a behavioral analysis where we put in relation the 5 measures that we have here of the brain with the 10 scores of sustained attention and fluid intelligence using partially square correlation analysis. So here you evaluate multivariate correlations between these 2 sets, brain and behavior, and what we found is that the highest correlation is indeed obtained for measures of decoupled functional connectivity. And you see here that regions of the brain that are in red positively correlate with the set of cognitive behavior that we have. So here it's multivariate, so you don't have one-to-one correlation values. But so the regions that you see in red, they include frontoparietal network that is already known, well known to be very important for fingerprinting from the seminal work of Finn of 2015. And these, so basically, interpretation why what you're saying is that connectivity in those areas is more decoupled when, for example, when sustained attention is increased, as it's a positive value in the behavior. So this concludes my presentation. So the take-home message is that graph signal processing revealed to be a very useful tool to integrate structure and function, and that we found out that this spans a gradient in the brain that has a behavioral relevance. And also more recently, we found out that there is a potential for task decoding and fingerprinting. So these measures of structure function coupling also uniquely identify subjects and tasks. And it's in the structure function decoupling in particular that you have the most important information to differentiate among different subjects. Thanks a lot, and thanks to my collaborators. The ones that worked mainly on this work are Alessandra Griffa and Dimitri Van de Ville. Thank you for your attention, and you can also come to my poster tomorrow and Thursday if you want to hear more details about this work. Thanks a lot.
So thank you very much, Julia. We are a bit short in time, so I'll just take one question and I will ask you to be concise so we can move on to next speakers. So the top-rated question for you is how was the cutoff between the low and high frequency bins determined, and what are the implications of this choice?
Can you repeat it, sorry? How?
How was the cutoff for the low frequency splitting determined, and the implications?
Yeah, great question. I mean, in the interest of time, I had to skip on that, but it's a very meaningful question. So, I mean, you have the choice. The cutoff, unfortunately, it's a parameter that you have to decide and that will affect, indeed, your analysis. So instead of picking just a number, like it was done also in the past, we decided to use a median split criterion. So we split the spectrum into two portions with equal energy. So this, you see, is the energy of the resting state functional signal projected on each of the harmonics, so the energy of each harmonics in the signal. And you see that the first ones, as expected, they have much more energy. And indeed, the cutoff, if you split the spectrum into two, it's in around 20, 30, depending on the data set. So it's very low. So basically, the first 30 harmonics, they explain the same energy as the last 330 in this case, because this example was on 360 parcellation, but yeah.
Okay, so thank you very much, Julia.
Thank you.
So our next speaker is Danny Bassett, which is well known, and from University of Pennsylvania, multiple departments. So Danny will give us a sort of like a refreshing break from GSP. So she will be tackling this idea of how we can integrate structure and function together from a different perspective using network control theory, which I think it's very nice to compare with GSP these ideas and to see how they can be, how they differ and what are the similarities. So could you just please play the video? Thank you.
Hi, everyone. I'm very excited to be with you all here today for this OHBM symposium and to talk about network control theory. I'd like to ask the question of what network control theory is, ask the question of whether it's useful, and then if it is, for what. So first, what is network control theory? So network control theory is a mathematical framework that determines which perturbations can drive the entire system to a desired state. It's typically applied to the study of the power grid, mechanical systems, air traffic control systems, and robotics. So here's an example of a little network of 4 different nodes where inputs U are placed inside a particular nodes to drive the activity of the rest of the system. And here's an example of what that might look like in a state space where the initial state may be at the origin and the final state driven by U may be somewhere else. Now, how would we take this idea and apply it to neural systems? Well, first what we would do is define a trajectory of a neural system to be the temporal path that the system traverses through diverse states, where a state is defined as the magnitude of neurophysiological activity across brain regions at a single time point. So, for example, a single TR in fMRI data or a single time point in some other kind of imaging technique. Then once that is set up, the controllability of the network refers to the possibility to manipulate network components to drive the system along a desired trajectory that is a set of states that culminate in a target state, which is often chosen for its functional utility, either in terms of task performance or in terms of healthy dynamics. Now, how does control theory differ from communication models? Well, first, they differ in their goals. So the goal of communication models is to capture the evolution of communication dynamics by using dynamical models, and then to characterize the process of signal propagation using graph theoretical and statistical measures. In contrast, the goal of control theory is to determine the control strategies that would navigate the system from some initial state to some final target state. So what are the inputs and the outputs of network control theory? Well, first, there are two different inputs to network control theory. The first is a map of the network, and so that would be a connectome or some sort of graph or some representation of the network that exists in the system. The second input is a model of dynamics, and we'll talk about different models of dynamics in a second. The outputs of the theory are a perturbation that has been designed to allow for some particular outcome. So let's talk about these three pieces in turn. First, the mapping of the network. So in a human, we would typically use diffusion tractography to map out the structural connectome in a human and use that as one of the inputs to network control theory. We could instead, however, also use track tracing in the macaque or mouse or EM in cellular level circuits. What are some advantages and disadvantages of different network maps? Well, first, I said that the common one would be structural connectivity. That has the advantage of allowing to model physical propagation of signals along white matter tracks. But it also has two disadvantages. One disadvantage is that it does not allow for propagation through local tissue where a white matter track has not been defined. So for example, when you have two gray matter areas that are next to one another and a signal could flow through the tissue, the structural connectivity may not show that connectivity. So one solution to that problem is to use what's called a touching matrix, which reflects the physical abutment of gray matter areas. And we've done that most recently in Carr et al. In a Journal of Neuroengineering paper. A second disadvantage of structural connectivity is that some states of the brain may privilege the flow of information along some tracks more than others, or in some directions more than others. And a solution to this disadvantage is to instead use effective connectivity matrices rather than structural connectivity. Though of course acknowledging that those effective connectivity matrices will only be as useful as they are well estimated. This is something that we have done recently in Scheid et al. 2021 in the Proceedings of the National Academy of the Sciences. I'll also note that multi-layer networks can be used to study control as well, so we could move beyond a single network and study multi-layer networks for the control of neurovascular systems, for example. Now, let's turn to the second input. I told you there were two inputs, a map of the network and a model of dynamics. So let's focus on the model of dynamics for a second. So first, the simplest model that we could use is a linear model that's also time invariant. And here's an example of what that would be, where x of t is the state of the system, A is an adjacency matrix, which could be the structural connectivity matrix, x of t is the state of the brain at time t. And then here's the control. U of t is the input injected into the system, and then B is a matrix that indicates which regions have input being injected into them. We could also move to linear time varying models. So here you have the adjacency matrix also being a function of time. That's particularly useful when you're studying effective connectivity matrices, where you may have different estimates of effective connectivity as a task is being performed or as an individual is moving through different dynamical regimes, for example, during a seizure. And then adding even more complexity, we can go to fully nonlinear models. But in each of these cases, what we can do is estimate what U of t is needed to allow for a particular state transition or set of state transitions. And once we've identified what that U is, then we can compute a energy associated with that change. And we do that by suggesting that the cost of control or the energy required for control scales quadratically with the input U. So that allows us to assess the energy required for these state transitions. Now, what are some of the advantages and disadvantages of these dynamical models? Well, first, let's start with the linear time invariant model, the very simplest one. This is nice because it's simple enough to provide analytical intuitions. You can actually derive things quite easily about it. It's been extensively developed in terms of its machinery or the methodology and tools available for many different control strategies. So there are lots of approaches that can be pulled from engineering into neuroscience. And it's a first approximation for nonlinear dynamics, often most accurate in the vicinity of the operating point. But let's move then on to the linear time varying models. How are these useful? Well, these are useful for studying context-dependent dynamics and for developing and understanding context-specific control strategies. They can build on some well-developed tools for estimating effective connectivity matrices. So for example, structural equation modeling, dynamic causal modeling, and transfer entropy. And they can take advantage of a lot of the machinery that has been developed for linear models, but just using it in sort of a piecewise linear manner. What about nonlinear models? So these are particularly helpful for scenarios when whole sectors of the state space are prohibited. So for example, not physiologically possible. And if you're interested in this approach and sort of an understanding of the accessible and non-accessible spaces of a dynamical system, I would highly recommend this paper from Cornelius et al., published in Nature Communications in 2013. Now I want to ask the question of, what if you have a lot of data from your system? Does that help you either choose which model to use or develop a new model? So if you have a lot of data, particularly stimulation data, then you can perform indirect predictive control using systems identification. Or you can use what's called direct predictive control. So how are those two different from one another? So first, indirect predictive control uses the data to fit a relatively low dimensional model, which you can then use in the control framework. In contrast, direct predictive control skips the fitting of the model altogether and simply uses the raw data inside of the control framework. And if you're unsure which of these approaches might be better for your particular scenario or your particular scientific question or data set, I recommend this wonderful paper from Vishal Krishnan and Fabio Pasqualetti called On Direct versus Indirect Data-Driven Predictive Control. It really helps you to think through which of those may be most relevant for your problem. So now that we've talked about the map of the network and the model of dynamics, I want to move on to the outputs. What is the output? How do we design perturbations? So there are several different outputs. Probably the simplest one is called the impulse response. So this is where this is a system's output when presented with a very brief input signal. So you just like ping the system and see what happens. So here's a bit of stimulation to a node that changes the activity of node I, but it also changes the activity of node J, which is connected to it. And if you sum up over all of these different responses from all of the different nodes, that provides you with the impulse response of the system. So for some models of dynamics, the impulse response can be solved analytically, removing the need for simulation and offering greater opportunity for mathematical intuition. The second output is a controlled response. So not just a ping to the system and see what happens, but a controlled response. This is the system's response to some controlling input U from some initial state X naught. For the linear system that I showed you earlier, the response will have two different components. The first will be the impulse response, so that was the simplest output, and then the second will be a convolution of the mapped inputs, VU, with the impulse response. The third kind of output that you can get from this approach is achieving a desired state transition. So because the map from the input to the response is known based on the model of the network and the model of the dynamics, one can design a mapped input, VU, to drive a desired response, XT. And also, I would note that there are many solutions that could drive that desired response. So it's interesting to think about which of those solutions may be relevant for the brain and what constraints may help the brain determine which to use. The fourth output is the controllability. So what is the controllability? Well, a system is controllable if there is a control input that brings our system from any initial state to any final state in finite time. And if you're interested in only a subset of initial versus final states, then you might study the associated control strategies like the average controllability or the modal controllability. Now I want to talk about the minimal energy control. And here is where we would design a control input, U of t, that would minimize the control energy and possibly some other factors to drive a desired response. So we're not asking, can you do anything to drive this desired response, but can you define a control input that minimizes the energy required for that transition? And the reason that that's interesting is that energy minimization is a common biological constraint, so something that the brain may be using anyway very frequently in deciding the state transitions that it engages in. But also there's a clinical reason to minimize the energy of the control, so as not to heat the tissue, for example. So with all of those outputs, so we've talked about the inputs, we've talked about the outputs. Now I want to ask the harder question: how do you know if you've made the right choices? So as with all modeling efforts, the answer is going to depend on the scientific question. I think that there are at least 3 different ways that we can think about the validity of the choices that we would make for the inputs and for the outputs. The first is descriptive validity. Does the network map and/or the model of dynamics match the data? If they do, then you have descriptive validity. What about explanatory validity? Do the outputs correlate with behavior, cognition, and symptoms? Does the theory provide candidate explanations of mechanism and of cause? If yes, then you have explanatory validity. And lastly, predictive validity. Does the framework predict the future response of the system to stimulation? Can it predict behavior, cognition, and symptoms? If the answer is yes, then you have predictive validity. Now, before closing, I wanted to mention a few recent methodological extensions that I'm particularly excited about in this space. One is moving from the control of activity to the control of functional connectivity. So not just can we push the brain into a particular state of activation, but can it be pushed, and is it pushed toward a particular state of functional connectivity? Secondly, we can separately assess transient control strategies, so control strategies that are very swift and short over short timescales, versus persistent control strategies, ones that have to expand over long time periods. We can also use those approaches to understand the control of different spatial scales of dynamics in the network. The third extension I want to mention is tuning the B matrix, which indicates where inputs are being injected into the system. You can tune that to account for regional variation in serotonin receptor levels. And this is really exciting work from Singleton and colleagues. Fourth and finally, I think it's particularly exciting to study brain states that are task relevant, so relevant for cognitive dynamics, such as those that are defined by beta weights of a general linear model applied to fMRI data. This is a really nice connection to cognitive function and understanding why that particular pattern of activation exists and how the brain got there and how it may move from one pattern of activation to another pattern of activation. The question spaces that the field is currently tackling include modeling cognitive control as a network control process. So, for example, this would include assessments of executive function and dysfunction and its development. Also relevant here are studies of the energy landscape modulation by either drug or placebo. Second open question space is using network control to better understand the functional consequences of altered brain structure in disease states. This would include studies of epilepsy, bipolar, and schizophrenia, and also relevant here are assessments of heritability of network control statistics. Third open space is using network control to better understand brain dynamics in response to tasks. So for example, studying the resting state versus visual processing, working memory, or also brain-computer interface control. Also relevant here are studies probing control energy as a marker of cognitive effort. And fourth and finally, an open question space is using network control to understand brain response to stimulation. So for example, from transcranial magnetic stimulation and grid stimulation in epilepsy. Relevant here are studies designing personalized stimulation paradigms that use the individual's own structural connectome and their own model of dynamics. So with that, I'd like to thank you very much for listening, and I'm excited to engage in the question and answer period.
Okay, so unfortunately we don't have Danny here, and as you notice, it was like a refreshing break from graph signal processing. But there are a lot of links, but unfortunately Danny didn't touch on to do the comparisons, but maybe already some of you noticed how these perturbation analysis, impulse responses can also be approached from a signal processing perspective. But still, if you have questions, please post them. Danny will log in and she will answer those. But now we move on to our next speaker, Sina, who is a PhD candidate at the Department of Biomedical Engineering and Melbourne Neuropsychiatry Centre at the University of Melbourne. And Sina's talk will be on how we can scale things up. Essentially, so far, all the networks graph that we've been talking on were region-wise graphs. But here, Sina will talk about voxel-wise graphs and to see how cool graph signal processing stuff we can do with on voxel-wise connectomes. So, take on.
Thanks so much, Hamid. So I would like to first thank the organizers and all the attendees to our early morning session. And I'll try to keep this talk a little bit shorter so that we could have a little bit of Q&A at the very end. So the original topic of the talk was spectral connectome analyses across modalities and resolutions, but I decided to change it to a more memorable or maybe simpler to take topic, which is graph signal processing for high resolution connectomic maps. So I will talk about graph signal processing applications to high resolution connectomes, and I will look into how different modalities and different resolutions relate into the topic of graph signal processing. So to give you an overview of what I'll be talking about, I'll give you a short introduction to graph signal processing, how we could define a graph Fourier transform, and graph signal filtering, followed by an introduction of how that relates to network neuroscience in general, how we could study brain network organization across structure and function and behavior, and how we could use the bold activation as a graph signal, and finally relate structural functional coupling to behavior using a GSP perspective. Then I'll move on to a specific take on high resolution connectomes and how that could be studied using graph signal processing, and how that could then be used to generate replicable and reliable behavioral biomarkers. And hopefully at the end of the talk, I'll leave you with a few concluding remarks as take-home messages. So the way I see graph signal processing is just a generalization of the traditional signal processing in which we have a time series, a discrete one if you say, and basically what signal processing could do through a Fourier transform is we take the signal from the time domain and convert it to a signal in the frequency domain. Now similarly, in a graph signal processing approach, and then we could also take the signal back from the frequency domain into the time domain whenever we want. We could also apply filtering to the signal if we want. Now, in a graph way of representing the signal, we have two components. Basically, we have a network, our graph, shown here in red, and then we have a signal that is basically some information that we have on top of our graph. Now, what graph signal processing mainly tries to do is to encode the information that we have over our graph into a bunch of harmonics, if you want to look at them, starting from our DC signal at the very left, our low frequencies where we could see at the middle that we have only a few switchings between the positive and negative parts of the signal, and then the higher frequencies where we have more switchings between the positives and the negatives. Now, much similar to traditional signal processing, in graph signal processing, we use a shift operator to compute this transformation from the signal domain in the space to the signal domain in the graph. And what we basically do is we do a decomposition, an eigendecomposition of the shift operator, and that helps us convert the information from the graph signal to the graph frequency domain. And similarly, we could go back from the frequency domain back to the signal domain. And one interesting take that we've seen in a couple of talks before in this session is that we could then use filtering to only take special frequencies of the graph signal and study those frequencies. And basically what we do is we take the signal from the spatial representation to the graph spectrum. We apply a filter that selects specific frequencies, if you will, and then we convert those filtered spectrum back to the signal spatial domain. So how does that all relate to network neuroscience, where we model the brain as a graph? So we have nodes and edges in this graph. The edges could be defined using either structural connectivity or functional connectivity. Earlier today, Katrina presented how functional connectomes could be used with GSP. Julia presented how structural ones could also be used. And now I'm also talking about a specific take where we use structural connectivity to define our graph, and then we use the functional MRI or the functional connectivity as the signal defined over that graph. And we've seen in network neuroscience research how these two closely relate. We know that the structure and function of the brain and their connectivity are closely related and correlated. However, the strength of the coupling or decoupling varies between different regions of the brain. And GSP, as you've seen, is a good tool to study this coupling. Now, the graph topology is used to define a network, and we assume that then the functional changes in the brain are some sort of transitions over this network. And we could then use filtering to divide this graph signal, if you will, to two different parts. A low pass filter will give us a low frequency mode, which is the part of the function that is closely coupled with structural topology. And we also get a high frequency mode, which is the part of the function that is more decoupled from the underlying structural connectivity. And works of Julia and her colleagues have shown that this approach of graph signal processing closely relates to previous work on functional connectivity gradients, and we see that regions of the brain that are more coupled are basically regions like the visual regions, basically the unimodal regions of the brain are more coupled to structure, and the higher order association cortices are regions that we see a more decoupling nature of activity. Now, where does high resolution connectomics fall into all of this? So recently we've been working on this way of representing brain networks without the need of defining a brain atlas. So traditionally, when we want to define a brain network, we have edges and the nodes are defined using brain atlases, and these atlases segregate regions of the brain into parts that are hopefully structurally and functionally segregated. However, we have seen that there are many atlases introduced in the past couple of decades, and there's a good concern about the replicability of study findings using different atlases. So we take a different approach in mapping brain connectivity, in which we use the high resolution brain imaging data that we have to define the nodes of our network. So basically the vertices on the cortical surface or the voxels in the subcortical regions and the cerebellum could be used as the nodes of the network, and we can define a connectivity matrix on top of that. We have provided publicly available code on how to map these matrices. The computational complexities sort of made us do it so that we could promote this in the future research. But similarly to the case in the lower resolution and high resolution, we could also apply graph signal processing. We basically use a graph signal shift operator that aims to describe how information diffuses over the network. We used the shift operator of the normalized Laplacian matrix, which is a normalized version of the Laplacian matrix that was described earlier, so that we assume that basically information from one region to other regions is distributed according to the strength of their connectivities. And we could use this shift operator, apply a component decomposition, and using the high resolution structural connectomes, these are the eigenmodes that we get. And there are a few different interesting observations in these harmonics that I would like to talk about. First of all, if you look at the very first harmonic, we can see, similar to previous work, it's kind of the DC signal of the network that we have, and it kind of shows in the normalized Laplacian matrix, it kind of shows the strength of connectivity in every vertex of the brain. And we then observe a very interesting pattern in that we see a separation in the eigencomponents between components that are mainly focused on the cerebellum and components that are more focused on the subcortical regions and the neocortex or the cortical surface. So this is kind of an interesting observation that we see this clear separation in the harmonics. Now, focusing on the cortical harmonics, Another interesting thing that we see is that we have a resolution-independent representation of the harmonics that are replicable across studies and across resolutions. So to give you an overview, these are the first 4 components of the high-resolution structural connectome graph signal harmonics. The first one is the DC that is always positive all around the brain. Then we have a left-right separation harmonic. After that, we have an anterior-posterior separation harmonic that is symmetric between the left and right hemispheres, and then we have another anterior-posterior separation that is now asymmetrically distributed in the left and right hemispheres. And this is actually a feature that we observe across graph signal processing or harmonic studies in the past. So this is the seminal work by Selen Atasoy, and we can see the left-right separation harmonic as well as the anterior-posterior separation. Also, Julia's work has shown the very same components, but in a lower resolution atlas-based network setting. And I would also like to say that this was also replicated in a recent work by Anjali Tarun, where they looked at a different type of high-resolution structural connectome in which every voxel of the whole brain forms a node of the structural connectome. And the main difference of their take and ours is that we only look at the cortical surface or the gray matter as the nodes of our brain, and we assume the structural connectivity is somewhat like an underlying wiring scheme that connects the whole gray matter together. Now, another thing that we did was kind of interesting. We first took the signal from the spatial domain, transform it to the graph signal domain, and we observed the energy distribution of the signal, and we observed that when we go to the high resolution, we have like 90,000 different nodes, so we get 90,000 different components. So we wanted to do this type of median split to see what proportion of the signal is represented in the lower frequencies and what proportion is represented in the higher frequencies, and we see this interesting power law distribution where only the first approximately 300 eigenvectors can describe half of what's happening in the resting state brain in terms of the energy of the bold signal. Now we use that to divide the fMRI signal into a low frequency and a high frequency, and we compute the state of decoupling of every single vertex on the brain. And we also see a map very similar to what we've seen earlier in lower resolution, a map that closely resembles the functional connectivity gradients, where in the higher order association regions are more decoupled and the unimodal regions are more coupled. This map is also representative of an earlier map that we derived that shows structural connectivity uniqueness. So basically, what regions of the brain are better at fingerprinting individuals, and we see that those decoupled regions in the structural connectome are regions that are better at also fingerprinting different individuals. Now, we wanted to move one step further and see if we could then use this information to derive predictions of behavior using this graph signal processing of structural function coupling analyses. So what we did was sort of a different take of behavior prediction. So we only took 40 individuals from the Human Connectome project. We tried to relate every individual's decoupling patterns with their behavior, and here we looked at fluid intelligence, and we observed that there's a significant association, if you will, between the decoupling patterns and how we could predict fluid intelligence from that. But our key question is whether that's replicable and whether that could be used to predict out-of-sample behavior. And what we did was we only used the sample training of these 40 individuals, and we then applied this to an out-of-sample generalization using the remaining 900 individuals from the Human Connectome project. And we interestingly see that the information that we were able to capture using only 40 individuals is generalizable to the whole population. And I think this calls for the fact that this analysis could actually be a promising way to produce replicable and reproducible results. Obviously, this is still biased towards the HCP data, and this calls for future research using this approach on other data sets to verify this. So as I promised, I would like to end my talk with a few take-home messages. So we observed that graph signal processing provides a computationally efficient method to analyze high-resolution connectomes, and these connectomes are usually hard to analyze because they're large in size, and graph signal processing is a great way of both dimensionality reduction and reduction of information into an interpretable information. We also noticed how graph signal processing techniques are relatively resolution independent and also crosstalk between different modalities. And finally, we noticed how GSP can be used to extract generalizable, reliable, and replicable behavioral biomarkers from diffusion imaging and functional MRI. I would finally like to thank my supervisor and colleagues, especially Andrew, who helped a lot in the whole process of developing this high-resolution connectomic pipeline, and also thank a special thanks to Thomas Bolton, who had a visit to Melbourne and visited us at Melbourne Neuropsychiatry Centre, and that the discussions with Thomas sort of derived all this idea of how to apply graph signal processing in a high-resolution structural connectivity setting. Thank you so much.
Thank you very much, Sina, for a nice talk. We are out of time, but I think we just take one question, and then there are a couple of other questions. You can then log in and answer the questions. So how much does the group consensus voxel-wise graph vary from subject-specific voxel-wise graphs? In terms of their harmonics, I think they're referring to.
So the difference of the...
So if you have the group-wise voxel-wise graph, and then you have subject-specific voxel-wise graphs, how do their initial set of harmonics quite differ?
Yeah,
I mean, how robust is the pattern you observe on the group-wise graph?
So there's a great deal of variability especially in the high resolution when we go to the individuals. The way we derived these harmonics was using a group average consensus structural connectome, but then we used every individual's functional activity to basically filter it using the normative group level connectome. I would imagine that it would vary a lot when we go to individuals, because previous work has shown that the individual variation in structural connectivity is enough to fingerprint every single individual in the population.
So great. Thank you very much again, Sina. And thank you for everyone else who attended this symposium. I hope you had something to take home. Yes, and
so I see there are very interesting questions posted on the app, so we'll try to answer that. But also, come to our posters, or in my regard, you can stop me in the corridors. I'll be very happy to talk about it. Thanks a lot.